cortical physiologist, so that's going to be experimental presentation, experimental data. But the more I'm coming from, not from Japan, but a little bit from the north of the north, and I'm going to give you a presentation, an overview of our research, which is in great need of interaction with the modeling people, because, which is focusing on, as the name says, on the interaction between long-range connectivity and local inhibitory microcircuits. I will present you a bit of an overview of different topics of the last, so say, decade, a little bit. In all mammals, we know that there is a kind of hierarchical structure of cortical connectivity from primary cortices via association cortices to premotor areas, and that this so-called feed-forward connection is reverberated by an equal amount of feedback connection, actually. And the posterior parietal cortex is a prototype model of this kind of intermediate position, because in all mammal studies so far, it is a multisensory hub, which is also then sending the fiber to the premotor areas and receiving presumably attentional feedback from the same areas. And this has been also shown to be true actually in rodents, where the PPC, which has expanded enormously in primates, is actually present, and that's the same type of connectivity, although it is like a really half millimeter, not even 1 millimeter, tiny stripe. Well, this long-range connection, of course, impinge on the function of the local microcircuit where they arrive, and we have seen yesterday super interesting data from Moritz on the canonical circuits in humans. I agree very much with him that we should stop talking about layer 2, 3, because physiologically these two layers are a bit different. But these colored arrows show that there are some noticeable short circuits in this sort of canonical circuit. Overall, the consensus is that the fact that the connectivity is layer and cell type specific is accounting for the fact that neurons in these different cell types, excitable cell types, display different functional receptive field properties, but they are also differentially sensitive to manipulation of the sensory environment. And we also gave some contribution to that. In a sense, the real richness, so to say, of the cortical circuit is actually not so much the excitatory cells, but the different varieties of inhibitory circuits, inhibitory cell types that target differential compartments of the target neurons, the dendrites, or the analogical spike input cam, or the soma, where the digital spike output is generated, and super interesting and molecular works by Jens Jelling Leffler here has helped us to characterize molecularly these different cell types, but also connect with the physiology that, and also offer the tool to have animals which allow to manipulate differentially these non-overlapping groups of interneurons, so to say. What is inhibition doing in all circuits? We know that inhibition is a crucial determinant of the temporal integration window of cells, is crucial for coincidence detection. This was a classical experimental work from Puil and Scanziani, where they have stimulated two input afferents in CA1, I think, and have observed the normal condition only when the second input fall in a very tight coincidence. There is a spike, which is this big downward deflection. But when instead the GABA A receptor are antagonized, then the temporal integration window becomes really way broader. On the other side, we know that timing is everything in the brain, and yes, there is so-called balance between excitation and inhibition, meaning that both during the spontaneous oscillation, but also during sensory evoked activity. We have also done, this was a work by Ver and Zador. We also did conductance extraction from intracellular recordings, and in many areas it is known that the inhibitory conductances come with a slight phase shift with slight delay and offering a temporal window, so to say, for the cell to spike. So this phase shift is basically confining temporarily the spike timing. Another role of inhibition is, of course, to be a determinant of the receptive field tuning properties of neurons. This is a picture taken from a nice review of Scanziani, showing that even if for inhibition has a broader, the inhibitory network has a broader tuning, which actually the experimental data show it has, for example, in case of orientation tuning, but also auditory tuning. Even in that case, since inhibition can basically, by, for example, hyperpolarizing, but also via shunting effect, move down the subthreshold response of the cell in relation to the action potential, this contributes to shape the tuning of the neuron. So this is so-called, this happens another way by exploiting the so-called iceberg effect. And indeed, in the last decade, this is a nice review, several studies with opto-genetics have shown that opto-genetic manipulation of the two main classes, dendritic targeting, somatostatin, or somatic targeting parvalbumin, can have, in different contexts, either so-called divisive effects, which can actually change the tuning, or subtractive effect, which cannot change the tuning. There is quite a controversy in the literature about that. Most of the work reports that in the cortex parvalbumin have a divisive effect, and somatostatin don't, but there are also data that show that it depends on the stimulus intensity, for example, because somatostatin cells are strong integrators. We also know that let's say, inhibitory networks are not only determinant of sensory responsiveness and receptive field properties, but they can also act as switches that change the brain, the network state. And this often happens via the fact that there is a so-called hierarchical organization of the synaptic organization of inhibition with inhibitory cell types which inhibit other ones, like VIP cell, which inhibit somatostatin cell. Actually, somatostatin cell also often inhibit the parvalbumin cells. And these so to say, the cells in the top position there are data showing that they can be modulated either by for the neuromodulation production, for example, the cholinergic brain basal for brain fibers do show activity with the calcium imaging that is correlating with the locomotion, and this we know is able in this way to disinhibit the dendrites, accounting for the fact that, for example, in the visual cortex, neurons respond better when the animal is running. But also, this type of disinhibitory motifs that we have seen yesterday have developed even further in the primate brain, in the nice presentation of Helbstatter. Can account for some other observation in the literature, for example, the fact that somatostatin cells get inhibited when the animal is actively whisking, and this is apparently due to the fact that the VIP cells are also contacted by feedback fibers, horizontal connectivity from the motor cortex in the bulbar cortex. And finally, it's embarrassing to show something like that after this amazing presentation. We know that the inhibitory circuits are also thought to be important for but also inter-area, so to say, communication by establishing a kind of a synchronization mechanism of the field potential. Also, in this case, note that it has been observed experimentally dissociation between the time phase of the inhibitor and the excitation, which is also locking the spiking, for example. But more research is really needed in this field of trying to understand how long-range excitatory inter-area connectivity affect the inhibitory microcircuit, and we try to do that in conditions which are physiologically, but also maybe pathologically relevant. So first, we will address basically what happens, how the a principle, so to say, of inter-area communication that we found between primary, so-called primary areas. Are they really unisensory? But also we should not forget that each primary area has, let's say, a sister one in the other hemisphere. Well, we know that also from working in macaques, for example, that in a primary area, it is possible to record this auditory cortex to record also cross-modal local field modulation, particularly in the upper layers, but that can also be able to affect, to some extent, the spiking of the cell in the spiking of the native modality, so to say. But however, in general, very few neurons spike in response to the other modality. So suggesting that this modulation, this cross-modal modulation, must be largely sub-threshold. And we did a simple thing. We just patched cells in light anesthetized, but also in awake animals, in mice, and we presented tones or white noise, and we observed almost always sound-driven hyperpolarizations that were accompanied by an upward field potential deflection, which were not preceded by an excitation. So these sound-driven hyperpolarization were revealed to be inhibitory, because when we extracted the conductance, we found a decrease of resistance and strong inhibitory conductance. But since we are experimentalists, we wanted to block also intracellularly the GABA system by putting pyrrhocortoxin and cesium, and this showed that sound-driven hyperpolarization disappears. Moreover, we also blocked extracellularly GABA with subepileptic dosages of GABA A and B antagonists, and as you can see here, the normally observed sound-driven hyperpolarization, these are grand averages, the black one, becomes significantly shorter when the GABA B is antagonized, starts later when the GABA A is antagonized, and basically disappear when both are antagonized. So this really says that these are inhibitory inter-area cross-modal interaction. And the fact that are inhibitory is also confirmed by the observation that the visual responses in primary cortex are actually reduced by a simultaneous sound, both at subthreshold and suprathreshold level. I'm saying that because, of course, an hyperpolarization could be due to either a withdrawal of excitation, but in that case, the input resistance of the membrane resistance increases, and this should actually increase the visual responsiveness. But the fact that that was a reduction instead indicate this is consistent with the shunting effect, so to say.
Can I ask you, what's the latency of this hyperpolarization after the start of the sound? So you see the kind of higher order recurrent or local?
We did, now I'm just reporting a summary of the study. We did a series of experiments, as I'm trying to do, where we did also transactions between the auditory cortex and the V1, and we found that they disappear. Mm-hmm. Moreover, we did also opto-genetic activation of the layer 5 cells in A1 to V1, so they come from acoustic cortex. And the latency are matching, so to say. The delay, if you activate the layer 5 cells artificially, and you make the difference with the sound activation in acoustic, it matches, let's say. So the idea is that the sound activates layer 5 cell in the acoustic cortex that then activate, and this is not shown here, but we have done multi-layer recordings and even in vivo patch showing that there is a tiny number of cells.
They are early. They're like 20 milliseconds after the start of the tone, right?
I don't remember now exactly, but I think in A1 it's 20 milliseconds, and then there is some other few milliseconds to reach V1. Yeah. But in V1, what happens is that a few layer 5-6 neurons are activated by sound, excited by sound. I don't show here the experiments, but we did also some really tiny injection of muscimol fluorescent in layer 5 and patched in layer 2-3 in the cortex. And we also checked for diffusion because that was a very tricky experiment, revealing that this is a form of translaminar inhibition within V1 coming from the deeper layers. And of course, an interesting candidate are the somatostatin cell we have here, GILA, that have classical work on that type of circuit. What could be the functional significance of this interarea inhibitory mechanism? It might be an attentional mechanism, might be a way by which the multiple areas kind of compete for access to the higher areas. We interesting found, and this is quite general, because by patching intracellularly, we found that the sound can inhibit also cells in the somatosensory cortex. And also a whisker stimulation done very carefully because we need to close the eye to check that the pH does not make a sound. But it also inhibits the visual cortex and the auditory cortex. Instead, visual stimulation like appearing of grating do not evoke apparently this interarea inhibition, because they do kind of nothing in the upper layers of this is patching of auditory cortex, but instead it focus like depolarization in the somatosensory cortex, in the barrel field. After that, the other work actually sort of confirmed and also characterized better this circuitry. Another group found that sound is actually activating some layer 1 inhibitory cells. And recently, the other work does show that tactile is evoking inhibition in the visual cortex via layer 6. So these modulator interneurons are often in layer 1 or layer super superficial, super deep. And this is a review where showing that basically there is kind of a consensus that the mechanism that we found is mostly happening during the day, whereas the other one, depending on layer 1 interneurons, is happening during the other part of the day cycle, so to say. It is also a bit provocative to see a paper from Carandini lab, which I still have to put in connection with our work, showing that acoustic stimuli of different character also induce, let's say, a bodily reaction, kind of startle-like, which is actually, he found, mirrored strictly by the membrane potential in the visual cortex. So, raising the possibility that, who knows, maybe these sound interaction are also taking into account some more motor somatosensory. Cross-motor interaction are complex. Okay, so we have described the circuit of inhibitory interarea inhibition, but we also were curious to explore how one sensory cortex of one hemisphere interact with the opposite hemisphere. In that respect, in the context of a study for the stroke, we recorded field potential here. This is the grand average by intrinsic imaging targeted the hind limb representation. So now we're in the somatosensory area. And when we blocked with muscimol the contralateral hemisphere, we unavoidably, in all animals, we've seen a huge increase of the response. So there is a very strong interhemispheric inhibition that is actually known to happen even in humans. When we apply the GABA B blocker, not the GABA. We have seen that this does not happen, and actually there is a trend for a reduction, unraveling some kind of also excitatory. So in the somatomotor cortex neuron, there is a very strong interhemispheric, tonic interhemispheric inhibition, because this is, compared to the previous study, this is a tonic form. And it was quite interesting to observe that in the vicinity of a stroke, because say this was a stroke study, this phenomenon basically disappears. So this was an intrinsic imaging. We positioned the forelimb, the hind limb representation. We did a photothrombotic stroke selectively in the hind limb, and then we documented the recovery also intracellular with patch clamp in the hind limb, which happens. But to our surprise, we found that when we repeated the same experiment, so we patched in the spared hind limb representation after, I think it was 2 weeks from stroke, we inactivated the other hemisphere, nothing happened. Irrespective of whether we stimulate the forelimb or the hind limb. So there is a loss of this strong GABA-B interhemispheric inhibition of callosal origin in the stroke periphery, and a possibility, we know that reducing inhibition is a good way to increase, to favor plasticity. In this case, plasticity does happen in terms of recovery of responsiveness. We also, very beautiful study of Matthew Larkin have shown actually that, yes, confirmed, actually before our, we confirmed that there is interhemispheric inhibition, and by activating the callosal fibers, he observed that the ipsilateral response to the forelimb reduced, and that this is accompanied by activation of some layer 1 neurons. At that time, he used organic calcium indicators, and we know that, and that was also GABA-B mediated, like we found. We know that a critical possible cellular mediator of that could actually be the neurogliaform cells, which are super interesting cells, big cells located in layer 1, which actually, via volume transmission, are able to inhibit with a single action potential hundreds of local neurons. It has to be observed that callosal interaction are not necessarily inhibitory. In the visual cortex, we have also shown that the net effect, the net tonic effect is actually excitatory, because they greatly contribute to binocularity, in particular the representation of the ipsilateral eye. But this circuit has to be, investigated better. I'm going a little bit fast because I want to give you an overview of our studies. We next want to ask ourselves what's happening actually in those areas where two senses are both excitatory instead of being antagonistic, like in the primary cortex. Meaning, what happens in the so-called truly multisensory areas, and a prototype example is the posterior parietal cortex. That work happened. This is just a brief, graphic sketch showing the function, the cellular correlate of multisensory integration in relation to our, perception and behavior. Let's imagine to have an audio-visual neurons that respond to the vision of a dangerous dog, but also to the sound that it emits. And when the dog is far away, maybe this neuron is spiking producing one and one spike. When the two sensory modalities are separated, and maybe 4 or 5 when they are presented together, meaning that there is a multisensory gain of like 400%, 500. When the animal becomes closer and closer, the unisensory responses become stronger, and due to the nonlinearities of the sub to supra threshold conversion, the gain becomes less and less. This probably reflects the fact that, in other words, the fact that multisensory enhancement is stronger when the unisensory information is weak, and being weak is also ambiguous, probably. These are the situations in which is more important. Studies, multi-electro studies from Wallace have shown that these multisensory neurons are mostly, if one grossly divides the cortex of a rat in sensory-dominated areas, the prevalence of multisensory units is more at the transition area, so to say. And that came at the time when Burkhalter did very nice anatomical studies, even in mice, showing that there are multiple association areas receiving retinotopic projections. And then he did a lot of work to really present evidence in favor of possible embryonic presence of also ventral and dorsal stream in the visual cortex of mice. Well, then we said, okay, why don't we focus on these tiny stripes between this ERL, between S1 and V1, where we should find the multisensory unit. And we actually, by intrinsic imaging, we targeted the tiny injection of calcium indicator. At the time we were using Oregon green, and we did found indeed an area where there is a kind of a salt and pepper presence of multisensory cells responding to whisker, to visual stimulation, or to both in green. We also did patch in this area where we found that in layer 2-3, in the upper layers, there is a strong multisensory enhancement. When the two stimuli are presented together, there is a strong. They have to be presented with a bit of a delay because the visual system, as usual, is terribly slow compared to the mechanosensory systems. But to our surprise, when we patched in layer 5, we found a tiny level of multisensory enhancement, and this is the quantification at subthreshold, at suprathreshold level. There is a strong, the multisensory enhancement index is way larger in layer 5, in layer 2-3, which is also a challenge for the classical canonical microcircuit, because how can it be that if layer 2-3 is the main driver of layer 5, that multisensory enhancement is not sort of transmitted to different layers? But when we also wanted to measure what do the inhibitory interneurons do, we found that they do integrate also quite little. In this case, we restricted our analysis to the paravalbumin cells by doing two photon targeted cell attached recordings. And this is an example showing that the scarce multisensory enhancement of the paravalvular spiking unit, whereas the regular spiking putative pyramidal cell with shadow patching instead showed strong multisensory enhancement. Then, of course, the question is, why are inhibitory neurons integrating less? But it is possible to think that this small multisensory enhancement might actually enable multisensory enhancement in the neighboring pyramidal cell, because when the two stimuli arrive together, if the inhibitory network is less integrating, the pyramid maybe are enabled to do it in a more effective way. And to kind of test for that, we helped a little bit artificially the GABAergic cells to fire a bit more. We did opto-genetic stimulation of the paravalvumin at the time of the multisensory test. So as you can see, the tactile stimulus reduced and the visual, so all the rest was reduced, but there was a stronger effect on the multisensory responses. So we found that multisensory integration in the posterior parietal cortical layer is cell-type specific, more pronounced in layer 2-3, and also more pronounced in excitory versus inhibitory fast spiking cells. And that we provided optogenetic evidence that this reduced multisensory enhancement of the inhibitory cells might be actually enabling multisensory integration in the neighboring pyramidal neurons. Of course, in the future, it will be very important to investigate better, because up to which level multisensory process in the parietal cortex, which is a kind of a bottom-up process in the way we study it, is affected by top-down influences. Because we know, I mean, anybody knows that posterior parietal cortex is linked to multisensory enhancement on one side, but also to attention. And of course, there can be a situation in which there can be the attentional demand can be convergent or divergent compared to the multisensory integration, particularly if the two stimuli come from the same source or from different sources. They might be, the brain need to either integrate or segregate them. There can also be situations in which one modality the animal must pay attention to the combination of modalities. For example, the classical example of the prey that needs to integrate maybe a slight sound, weak odor, shadow, in order to guess the presence of a predator. So there are situations in which the attentional demand is convergent with multisensory integration, but there are also situations where instead one modality is actually disturbing. So in the future, it would be nice to investigate how this network behave in these two types of situations, so to say. And there are already some initial report for addressing this question. We went then a little bit in the cortical hierarchy. We moved a bit sort of up. And but tried still to keep it simple enough to be able to do mechanistic work. We have focused in this third part on acoustic responses, in particular to ultrasound responses. You know that animals use ultrasound rodents for vocalization, which is social importance. We found that there is a specific region of the prefrontal cortex, the orbitofrontal areas, which receives a direct monosynaptic connection from the auditory association cortex. We did an anatomical study that now we have developed even further. And we found that there are acoustic responses also in the, this is an example, this is the grand average, in the orbitofrontal cortex, which are strongly reduced by chemogenetically manipulating selectively this connection by using a retrograde virus, which expresses the Cre and then injecting the flux, the chemo inhibitory in the target region. Then we injected the CNO, the chemo activator, in the recording pipette. We found that we confirmed that this is a functional. We also confirmed that these acoustic responses are very sensitive to dopamine. They're basically cancelled by apomorphine, but also by neurosteroids, which are molecules which are important for, say, in psychiatric conditions. So I think I'll skip the next one. And then we focused on a phenomenon which I thought was not present in a rodent in this extent, the sensory gating. Sensory gating is an expression of, let's say, many people say it's the inhibitory side of attention. Acoustic, there is a very simple test that consists in presenting two sounds interleaved by 500 milliseconds, and can be done with was done in the 80s, actually, in the clinical context with electroencephalography. And the remarkable observation was that in the prefrontal areas there is a strong suppression of the second, which is absent in psychotic patients, in schizophrenia, and also, interestingly, in the first degree relatives of patients, the healthy ones, suggesting that it is then further study worldwide, basically, I've confirmed that it's one of the most reproducible so-called endophenotype of schizophrenia, or better said, risk for psychosis, because the same sensory gating deficit, which is shown here in red, is also present in, for example, bipolar, psychotic, but not in the non-psychotic. Well, quite surprisingly, we found when we studied this connectivity, this long-range connectivity between association cortex and orbitofrontal, we found basically no gating in the acoustic association cortex, but instead a strong one in the orbitofrontal cortex, though the response to the first became really long. But the second was almost cancelled. The fact that there was no gating in the input area raised the possibility that this could be due to local recruitment of GABAergic cells and not to reduced excitation. in order to confirm that, we basically did we used the field potential on purpose because in this case we wanted to do something which is similar to the clinical test, which is the EEG, and we injected locally a mixture of the subepileptic mixture of GABA A and GABA B antagonists. This is one example. This is the grand average, and we found that the second wave come clearly back without affecting the first. So this is the saline conjunctive control, and even just GABA B, which is most expressed at the dendritic level, is able to reactivate ungate, so this is the second response. We then performed, we wanted to know, okay, this suggests that the local GABAergic circuit are gating the second response. Which cell type? We then did opto-genetic inhibition of the two main effector classes, so to say, of in the parvalbumin cell. And when we did that, we found that basically both responses increased similarly, then depending on in line with the kind of diffusive effect of inhibitory cell, depending then on whether one measures the peak-to-peak or the baseline to peak response. There is basically not a strong effect on the ratio, which is what is clinically relevant. When we instead with the same manipulation on the somatostatin dendritic targeting cell, we found that there is a reduction of the first wave, so not an increase, but an ungating of the second, meaning that basically that was actually really inducing a complete sensory deficit in sensory gating. So the ratio was way higher. And so these data are in line with the hypothesis that the somatostatin interneuron are the most important mediator of this phenomenon, which is profoundly altered in the psychotic prefrontal cortex. To my surprise, there was no mechanistic studies about something so simple to be studied, but and I think this is exactly what is fascinating, because a postdoc of mine told me, like, sensory gating is the smoke, and schizophrenia psychosis is the fire. So we are studying the smoke, but if we know the mechanism of the smoke, maybe we also can know the mechanism that drives the fire. Well, since dopamine is super important for psychotic states, and is when the neuroleptics actually are used to treat patients, we wanted to test what happens when we counteracted dopamine. And to our surprise, we found that neuroleptic consistently induced a deficit in sensory gating, yes, by reducing the first and increasing the second, which is exactly mimicking the effect of the blockade of the somatostatin neuron. So this raises the possibility that dopamine is facilitating gating via the dopamine, the somatostatin neuron. If that would be the case, then photostimulation of the somatostatin interneuron should counteract, bypass the neuroleptic effect. And we actually did this experiment. So we did an experiment in which we injected neuroleptics, which induced a deficit in sensory gating, as you can see in the grand averages or in the examples. The two waves are kind of similar. But then we photostimulate the PV cells. This is nothing happens basically. But when we instead photostimulated the somatostatin neuron, the second wave became significantly smaller, normalizing also the sensory gating ratio. This is a bit of an enhancement of the. So nothing happens here to the second wave, which is the gated one, the one that was ungated by the neuroleptic, but the neuroleptic effect was significantly recovered, and also the ratio, by photostimulation of the somatostatin cells. We did also other experiments, but in summary, I would like to highlight that this this series of this work, which is now under revision, is allowed us to propose a microcircuit hypothesis on how the gating happened. Basically, our hypothesis, which is consistent with the data, is that the pyramidal cell fire for a sustained time to perform a temporal integration, because data from the literature know that they express more an MDA receptor, which allow to longer tau off of the unitary EPSP. And then these allow them to inhibit at the dendritic level the pyramidal cell, so that when the second response comes, the second input comes from auditory cortex, the neurons are already inhibited, so to say. And the effect of GABA B is also in line with the predominant role of the somatostatin cell, because the GABA B is mostly dendritic. Of course, the fact that the GABA B was also antagonism was also able to ungate the response does not allow to exclude that this might actually happen at the level of the axonal terminals coming from the acoustic cortex, because the axon also express presynaptic GABA B. Terminals, able to modulate another electrophysiological phenomenon, which is called mismatch negativity. Basically, it is a change in the response which happens when the animal is, after being exposed to a regular sequence of stimuli, there is an unexpected stimulus. Schizophrenic patients also display a deficit in mismatch negativity, and maybe it's not by chance that the somatostatin cells also are affected here. All in all, there are some, let's say, this model that we propose is in line with the idea that the somatostatin cell operate more in a subtractive way compared to divisive inhibition. And of course, you understand that to change the ratio, subtraction is what is needed, because if you have a multiplicative effect, the ratio will not change. On the other side, I think it's for our working hypothesis is quite crucial for stimulating pyramidal cell and then recording intracellular from parvalbumin or somatostatin. And also in this case, it was quite clear that due to the longer unitary EPSP of the somatostatin, which is attributable to the higher expression of the NMDA, this cell type is able to integrate better in time. And finally, the fact that dopamine is facilitating mostly the somatostatin neurons is also in line with observation from Goldman-Rakic, which was showing a differential effect on the inhibitory current from a pyramidal cell to, from fast spiking or putative somatostatin neurons. So there was an opposite modulation of dopamine, diminution of inhibition from fast spiking and increase from the putative somatostatin. A data result which have been also recently confirmed in the hippocampus. One slide of this, about a summary slide, an invitation maybe to read a work which is, I think, is the most original that I did, but and for that is less quoted, which is about another long-range interaction between and also with cross-modal plasticity, how sexual stimulation, in particular vaginal cervical stimulation, affect the activity in the olfactory areas. One can think that this is maybe not so relevant, but it is relevant because in in Nature in the 50s, there was a work showing that the so-called Bruce effect is a dramatic phenomenon that shows basically that if a pregnant female smells the odor of the mating male, the pregnancy continues. But if she smells the odor of another mate or another non-mating male, another male, in 80–90% there is abortion. And that this is this kind of, because if lidocaine activation of this idiot right after the mating is preventing this memory. Then I thought that this tiny idiot, of course, there was a specific person that was an expert on this circuit, must also receive not only the olfactory information about the identity of the male, but also the mechanosensory input during the coitus, basically, in order to connect the to form associated memory. That was my plan, so to say. I'll skip all of this, and I will just go to the, that's in iScience. We proposed in the end, by a series of intracellular recordings, doing artificial vaginal cervical stimulation in estrus and the estrus, we proposed the microcircuit hypothesis on how vaginal cervical sexual input is represented in the olfactory circuit. And the surprising thing is that we found that artificial vaginal cervical stimulation done in a way that actually mimics the coitus is inducing inhibition, again, of activity of the mitral cells. And this happens only in estrus when the female noradrenaline via locus ceruleus, which is activating the granule inhibitory cells, which are primed, because you know that the inhibition here happens by the endodendritic synapses, and these the endodendritic synapses are sort of primed to be more excitable specifically during estrus, because during estrus we found that the spontaneous activity is biased towards burst firing, and we know that burst firing, by previous work on from actually Nathan Urban, Sackman also, by backfiring, burst firing is able to potentiate the endodendritic inhibition. I think one thing that was very surprised is the total lack of any information on how the sexual input is represented in the brain. The total lack, compared to like hundreds. Thousands of work on visual symbolism. So I think there is a big need to open this here. Quite interestingly, Michael Brecht has also observed that the inhibitory fast spiking cells are also sensitive to the estradiol levels, and they become way more excitable in the basal cortex during estrus. So, yeah, that was a bit of a presentation. I understand it's heterogeneous, but I think the feeling that I have is that we are often getting pictures of disparate phenomena, and it would be nice to sometimes model some of them to have a bit more integrate with the cortical microcircuit function a little bit better. So these are the collaborators. Thanks a lot.
Bit late, but I think we still have time for a couple of questions.
I was wondering about the sensory dating, if you look with the similar ADHD.
In ADHD?
Yeah, I'm thinking there's also like similar problem there? Or?
Yeah, yes, indeed, there is also a problem. Yes, there is also deficit in autism and ADHD. The idea somehow is that the sensory gating deficits accompany condition where these executive symptoms are prominent, because we shouldn't forget that in schizophrenia also part of the so-called negative or cognitive symptoms are autism-like. So in my opinion, these are all clinical situations in which, but also not only in my opinion, in which there is a deficit of the inhibitory function of the prefrontal circuitry, which express as so-called disexecutive symptoms. But it is also interesting that the relation with psychosis, which is the other side of schizophrenia, because again there is a sensory gating deficit also in the psychotic bipolar, but not in the bipolar which are non-psychotic. So the consensus is that the sensory gating deficit is also expressing a vulnerability for the risk of psychosis, so to say, which is up to which extent this is linked to the, let's say, attentional deficit has to be, but maybe they are both mechanistically linked.
Yeah. Okay. So your multisensory, shall we say, modulations, especially in layer 5, 6, and in layer 2, and even layer 1, as you show, have, shall we say, local mechanisms, as you have proven. But can you exclude the influence of what Ted Jones calls the matrix, thalamic matrix neurons, which actually end up in layer 2 and to some extent layer 1 and layer 5, layer 6. So
absolutely not.
And is there's not much known about their detailed connections, and not much known about when they appear in latency when you stimulate. But
absolutely, it's impossible to forget like work of people like Schaeffer that always support the idea that the cortical communication is mostly mediated via associative thalamus, actually. But what I can just say experimentally is that we did very careful transaction experiment between, for example, visual primary, and when we did that, we also controlled that after the transaction, the evoked potentials in the two respective areas were not changed, indicating that probably we didn't severe thalamo-cortical let's say the primary thalamocortical projections. But I think there is also some recent work actually showing that no, absolutely no, I cannot. But the fact again that we found that these sound-driven hyperposition were really GABAergic in nature, were suggesting that there was a local origin. So whereas the thalamic mechanism should express more as a withdrawal of excitation, right? Yeah, because the thalamus is providing, so it should be, but then if you have withdrawal of excitation, again, the neuron, the input resistance of the cell should become higher, and so the visual responsive should become larger when given simultaneously sound, which they don't. But in a awake behavioral context, because this was a study in the light anesthetized, but also in animals, so maybe it's different.